knows a lot about the science stuff. Professor Dave explains.
For much of this series, we have been examining the innate immune system. Before we move on to the ever so captivating adaptive immune system, we have to get a closer look at some of the cell types we have been mentioning throughout the series, each of which will get their own tutorial. First up, let's talk about macrophages. Macrophages are specialized, long-lived phagocytic cells of the innate immune system. They play a pivotal role in recognition, phagocytosis, and degradation of cellular debris and pathogens. Macrophages also participate in tissue development as well as repair and resolution of inflamed tissues. Additionally, these cells present antigens to T cells, and they induce the expression of co-stimulatory molecules on other antigen-presenting cell types, which ultimately initiates the adaptive immune response. Furthermore, they are crucial in the initiation of inflammation by releasing pro-inflammatory cytokines and chemokines, which in turn recruit other immune cells to inflammation sites. Macrophages were first discovered late in the 19th century by Ilya Mechnikov, and they are evolutionarily conserved phagocytes found in vertebrates. Initially, scientists proposed that all tissue macrophages originate from the circulating blood monocytes, which continuously migrate to different tissues and differentiate into macrophages. However, recent studies have overturned this prevailing paradigm, as several independent groups reported the results of genetic lineage tracing studies in mice. They provided strong evidence that resident tissue macrophages are in fact established during embryonic development and persist into adulthood independently of blood monocyte input in the steady state. Despite the dramatic progress made in the field of macrophage research, the dichotomy between embryonically derived and adult-derived macrophage populations does not fully encapsulate the complexity and diversity found among tissue macrophages. Depending on the tissue microenvironment, macrophages exhibit plasticity in terms of phenotypes and function. Macrophages can infiltrate various tissues under inflammatory or non-inflammatory conditions. They can assume different functions and phenotypes according to the cues they receive from the microenvironment. Broadly, macrophages can be divided into two distinct subtypes. 1. Classically activated, or M1 macrophages, and 2. Alternatively activated, or M2 macrophages. Let us discuss how these two subtypes of macrophages are formed. In vitro, M1 macrophages are polarized by Th1 cytokines, such as colony-stimulating factor, tumor necrosis factor alpha, and interferon gamma, either alone or together with lipopolysaccharide from bacteria. M1 macrophages express pro-inflammatory cytokines such as interleukin-1 beta or IL-1 beta, IL-6, IL-12, IL-23, and TNF-alpha. In contrast, M2 macrophages are polarized by Th2 cytokines, such as IL-4 and IL-13, and produce anti-inflammatory cytokines such as IL-10 and transforming growth factor beta, or TGF-beta. It is important to note that macrophages can adapt special tissue microenvironments to polarize specific phenotypes such as tumor-associated macrophages, or TAMs, and adipose tissue macrophages, or ATMs. Each phenotype of macrophages has a relatively specific expression of some cytokines, chemokines, toll-like receptors, or TLRs, and matrix metalloproteinases, or MMPs. The M1-M2 paradigm is unfortunately not sufficient to address all polarized phenotypes that macrophages can assume. This is because, depending on the stimuli, they have the ability to modify their polarization status. For example, macrophage phenotype can change during tissue repair, switching from pro-inflammatory phenotype, or M1-like, to an anti-inflammatory phenotype, or M2-like. Also, different polarization and activation markers of M1 and M2-like macrophages can coexist in tissues at the same time. Therefore, it is extremely important to be aware of the cellular and functional plasticity while studying macrophages in vivo, such as in diseases. M1 macrophages express high levels of major histocompatibility complex class II, or MHC2, CD68, as well as CD80 and CD86 costimulatory molecules. M2 macrophages express low levels of MHC2 while featuring expression of CD163 and CD200R membrane glycoprotein, as well as high levels of MGL1 and MGL2, which are members of the macrophage galactose type C lectin family. Again, macrophages are phagocytic in nature. Other notable phagocytic cells are granulocytes, consisting of neutrophils, eosinophils, basophils, and dendritic cells. When a pathogen enters the host and begins to replicate, it is immediately recognized by one of the macrophages. Membrane protrusions surround the pathogen, absorbing it into the phagosome, which is formed by the fusion of cell membranes. Most pathogens gain access to the host through the respiratory system, gut mucosa, skin lesions, or the urogenital tract. Macrophages are well known to populate submucosal tissues, and therefore they are usually the first defensive cells to encounter harmful pathogens. Phagocytosis is initiated when certain phagocyte receptors, usually carbohydrate or lipid structures specific to pathogens, interact with the pathogen surface. A wide range of phagocytic receptors and a variety of signaling cascades can be activated during this process. After being taken into the phagosome through one of several methods of receptor stimulation, the pathogens are eliminated by oxygen-dependent and oxygen-independent attacks. Within the phagosomes, NADPH oxidase produces reactive oxygen species, and phospholipase A2 produces free fatty acids. These molecules are extremely effective in killing the pathogen. This is followed by actions of myeloperoxidase, or MPO, in addition to various hydrolases and lysosomes, such as azurophilic granules that fuse into the phagosomes, which ultimately degrade pathogen fragments. Next, the proton ATPase pump reduces the overall phagosomal pH, thereby activating either a strong oxygen-dependent sterilization system, mainly through halogenation, or an oxygen-independent sterilization system using lysosomal enzymes. In addition to pathogens, macrophages also remove apoptotic cells in the same manner. Despite their phenotypic plasticity, macrophages have been of immense interest to researchers as being valuable therapeutic targets in various disease settings. Several elegant strategies have been pursued with the aim of functionally manipulating macrophages. Of these, their re-education and total depletion can be considered some of the more successful strategies. Macrophage re-education has been used in the field of anti-cancer therapy, in which M2-like pro-tumor TAMs are reprogrammed as M1-like macrophages, endowed with anti-tumor functions. Macrophage depletion is a more extreme approach used to block macrophages, thus directly killing them off. Depleting antibodies such as anti-colony stimulating factor 1 receptor, or anti-CSF1R, or molecules exerting a specific toxicity against macrophages, such as bisphosphonates like clodronate and zoledronic acid, as well as trabectedin, have been widely used. With macrophages covered, let's move forward and look at some more immune cell types.
Thanks for watching. Subscribe to my channel for more tutorials. Support me on Patreon so I can keep making content. And as always, feel free to email me, ProfessorDaveExplains@gmail.com.